Welcome to BioTales, the show where we dig into the stories behind biotech breakthroughs. And today, wow, we've got a headline that really jumped out. Bayer just bet up to $1.3 billion that Kumquat's KRAS G12D drug can crack one of cancer's toughest codes. I mean, $1.3 billion, that's a pretty massive vote of confidence, isn't it? Really suggests something special is going on at this company, Kumquat.
Now, before we really jump in, have you ever actually tasted a kumquat, the fruit?
You know, it's funny you ask. I have. Well, they're quite surprising.
Right.
Small, yeah, but then this incredible, bright burst of flavor. Really unexpected.
Precisely. Picture that tiny citrus fruit for a moment. Golden, kind of glossy, and so resilient it thrives even in harsh conditions, and then, boom, this unexpected burst of sweetness.
Yeah.
Now, imagine a group of scientists naming their entire company after that very fruit. They chose the name because they believed their medicines should mirror those exact qualities, thriving in the toughest tumors and delivering an unexpected burst of hope. This, listeners, is the story of Kumquat Biosciences. Our journey today winds through the incredibly complex world of RAS genes. These genes have, quite frankly, haunted oncologists and drug developers for decades.
Absolutely.
We'll follow the scientific team that courageously tackled the undruggable KRAS oncoprotein, and then we'll see how they're looking to turn small molecules into what they're calling immune system whisperers.
Two really distinct but potentially synergistic approaches.
So settle in. This tale is truly part science, part startup saga, and yes, a tribute to a humble fruit that sparked an entire mission.
Okay, so to really get Kumquat, especially that huge Bayer deal, we need to talk RAS proteins. Anyone listening who's in oncology knows these names. They're like molecular switches, controlling cell growth. Normally, they cycle between off, which is GDP-bound, and on, TTP-bound, telling cells when to grow and divide.
Well, what happens when they get stuck? Stuck in the on position?
Uh-oh. That's where the trouble starts. Tiny errors, specific point mutations in the gene, at places like codon 12, 13, or 61.
Just small changes in the code.
Exactly. They can cause the switch to get jammed permanently on. We call that constitutive activation. This leads to uncontrolled cell growth, proliferation, basically cancer. And KRAS specifically is a major player here. It's one of the most mutated oncogenes found in maybe 25, even 30% of all tumors.
Wow, that's a lot.
Yeah, especially early on in things like pancreatic cancer, colorectal cancer, and non-small cell lung cancer.
And I read that one specific variant, G12D, that's a glycine swap for aspartate at codon 12, is particularly nasty.
It really is. It drives something like 37% of pancreatic cancers.
Which has just a terrible prognosis, very low 5-year survival rate.
Tragically low. And G12D also shows up frequently in colorectal and lung cancers, too.
So for years, scientists called KRAS undruggable. Why was it so hard to target?
Well, for a couple of key reasons. First, unlike a lot of proteins drugs target, KRAS has this remarkably smooth surface, almost featureless.
Not many nooks or crannies for a drug to bind to.
Precisely. And second, it grabs onto its natural partner, GTP, that cellular energy molecule with incredibly high affinity. It just holds on super tight.
So it's hard for a drug to compete and knock the GTP off.
Extremely hard.
Yeah.
It really took, a combination of structural insights, clever chemistry, and just sheer persistence to even start figuring out how to drug it. The first real sign of hope, that first crack in the fortress, came back in 2013. This was down to some brilliant work by two chemists, Eli and Pingda.
That's right. They developed the very first covalent inhibitor that was designed specifically for a KRAS mutant, the G12C variant.
What does that mean exactly? How does it work?
So think of most drugs like a key fitting into a lock. They bind, do their job, and then they can leave. A covalent inhibitor is different. It forms a permanent, irreversible chemical bond with its target protein.
Like supergluing the key in the lock, permanently jamming it.
That's a great analogy, yeah. It locks it up. And crucially, for G12C, there's a specific amino acid, a cysteine, that's present in the mutant protein but not the normal one. It acts like a unique handle. The Lu and Ren's inhibitor was designed to grab onto that cysteine handle and permanently lock KRAS G12C in the inactive state. Very specific, very effective.
Wow. Okay, so that G12C discovery was huge then. It proved KRAS wasn't completely undruggable after all.
Absolutely monumental. It directly led to the development of the first approved KRAS drugs, like sotorasib and adagrasib. And it's really significant. G12C only accounts for a certain fraction of KRAS cancers.
Right, so what about the others, like the big one, G12D?
Exactly. G12D and other mutants were still left out in the cold. And critically, G12D doesn't have that cysteine handle. The mutation replaces a glycine with an aspartate. No cysteine means no easy anchor point for those first-generation covalent drugs. So a massive unmet need remained.
And Liu and Ren saw that gap, didn't they? They saw it as the next major challenge.
They did. And you have to understand, these two aren't just average chemists. Yi Lu and Pingda Ren, they've worked together for, over 15 years? They have this incredible track record.
Tell us about that.
Well, back in 2007, they co-founded a company called Intellikine, developed some really important PI3K and mTOR inhibitors. Takeda ended up acquiring them.
Okay.
Then, in 2012, they launched Wellspring Biosciences. And that's where they did the pioneering covalent chemistry work for KRAS G12C. Wellspring later spun out Kura Oncology, which went public in 2015.
So there's a pattern here. They identify a tough problem, build the science, get it to a certain point, and then move on to the next big thing.
Precisely. A pattern of serial innovation, you could say. They seem drawn to the really hard foundational discovery work.
So fast forward to 2018, Liu and Ren are ready for a new venture. They'd cracked G12C, but knew the job wasn't done.
Right. They saw two key things. First, the urgent need for drugs against mutants like G12D. And second, this growing realization that even powerful immune therapies weren't working for everyone. Too many patients were being left behind.
So their vision was ambitious, to say the least.
Very ambitious. They wanted to build a company that could tackle both combined targeted therapy, like hitting KRAS directly, with immuno-oncology. And they wanted to do it using small molecules, aiming for deeper, more lasting responses in patients.
And that brings us back to the name. They needed something that captured that spirit resilience, hitting hard despite challenges.
And they landed on kumquat, that small, tough fruit with the surprising sweetness. It's actually a perfect metaphor for what they wanted their medicines to be.
It really is. So Kumquat Biosciences was quietly incorporated in San Diego, November 2018. They officially launched in 2019, and with Liu and Ren's reputation preceding them, investors were definitely interested.
I bet. That track record speaks volumes.
Absolutely. They raised over $100 million pretty quickly. Top VCs like Obamed, Sequoia China, Ecor One, Lilly Asia Ventures, Roche Venture Fund, they all jumped in.
That's serious backing. So what was the immediate plan with that capital?
Build two core platforms. Platform one, push the boundaries on covalent inhibitors for RAS, going after those other mutants. Platform two, develop small molecules that can modulate the immune system.
Okay, let's dig into that first platform. We talked about covalent chemistry for G12C, grabbing that cysteine handle. But G12D doesn't have the handle, right? It's glycine to aspartate. No sulfur atom for that classic covalent bond.
Exactly. No easy hook. That was the G12D challenge in a nutshell. So the old trick wouldn't work.
So how did Kumquat tackle it? This is where the real ingenuity comes in, isn't it? Instead of trying to force the old chemistry, they started exploring different types of interactions, sometimes called pseudocovalent. They designed molecules that could maybe exploit temporary pockets that only appear fleetingly on the protein surface, or perhaps induce a change in the protein shape to create a new binding site.
So finding a way to make the drug stick really tightly, even without that permanent chemical bond.
Sort of, yes. Or achieving similar irreversible effects through clever design. It's about designing molecules that bind so well or trap the protein in an inactive state so effectively that it mimics the long-lasting effect of a covalent bond, even if the chemistry is technically different.
Like finding a secret way to jam the lock, even if there's no obvious keyhole. And they weren't just focused on G12D, right? They were thinking broader.
Right. Also looking at pan-KRAS inhibitors drugs that could potentially hit multiple different KRAS mutants at once. That would be a huge advantage.
Okay, let's talk pipeline specifics for this platform. They have KQB365.
Yes. KQB365 is a covalent inhibitor, but this one targets both KRAS, G12C, and another mutation, G12S. They started phase 1 trials back in February 2025 for advanced solid tumors.
And the trial design.
Patients get weekly infusions. They're testing it alone and also combined with cetuximab, which is an EGFR inhibitor. The goal is standard phase 1 stuff, find the right dose, check safety, and get those first hints of whether it's shrinking tumors. It builds on their G12C expertise but expands it.
Makes sense. But the one that really made waves is KQB548, right? The G12D inhibitor.
That's the one. This is the program targeting the mutation that people called undruggable for so long. Kumquat got the FDA green light for clinical trials in July 2025. This was a massive milestone.
And it immediately led to something even bigger.
It did. Just a month later, August 2025, the news broke about a huge partnership with Bayer, centered specifically around KQB 548.
Wow. Tell us about that deal.
It's a major validation. Kumquat runs the initial phase 1A trial, figures out the dosing and early safety. Then Bayer takes over all the later stage development and hopefully commercialization.
And the financials must have been significant.
Very significant. Up to $1.3 billion in potential payments for Kumquat. That includes the upfront money, plus development and sales milestones down the road. And they get tiered royalties on sales. Plus, they smartly kept an option for profit and loss sharing here in the U.S.
So a huge potential upside for Kumquat. And for Bayer. Why was this so attractive?
Well, KRAS mutations are in, a quarter of all human cancers, and G12D is a huge chunk of that, largely unaddressed. Partnering with Kumquat gave Bayer immediate access to a potentially best-in-class approach for this critical target. It plugged a major gap in their oncology pipeline.
So this deal wasn't just money, it was a massive stamp of approval on Kumquat's G12D science.
Absolutely. It sent a strong signal. Kumquat's chemistry expertise is generating real high-value assets that Big Pharma wants. It really positions them as a leader in the KRAS space.
Okay, so that's the KRAS platform, really impressive progress. But Kumquat was also building a second platform in parallel, right? Immuno-oncology, but with a twist.
Exactly. Not just antibodies like checkpoint inhibitors, but small molecules designed to modulate the immune system. It's a less crowded field, potentially transformative.
And the goal here is different from directly hitting the cancer cell.
Right. The idea is to use these oral small molecules to activate the body's own innate immune sensors, or maybe even reprogram certain immune cells within the tumor. The big picture goal is to turn immunologically cold tumors into hot ones.
Okay, cold versus hot tumors. What's the difference there?
So a cold tumor is basically one that the immune system is ignoring. It doesn't have many cancer-fighting T cells inside it. The immune signals are weak. Maybe the cancer cells are hiding their identifying markers.
So the immune system just doesn't see it or can't attack it effectively.
Precisely. And often the area around the tumor, the microenvironment, is actively suppressing any immune response. You have cells there telling the T cells to back off. That's why standard checkpoint inhibitors often don't work well in cold tumors. You need to heat them up first, make them visible and vulnerable to the immune system.
And small molecules might be better suited for that than antibodies.
That's the hypothesis. Small molecules can generally penetrate tissues, including dense tumors, more easily than large antibodies. They can get right into that suppressive microenvironment. And you have more flexibility in combining them with other treatments, like targeted therapies or checkpoint inhibitors.
So how would these small molecules heat up a tumor? What's the mechanism?
The idea is they could activate these innate immune sensors that kicks off a cascade. Immune cells, like dendritic cells, mature. They start producing signal cytokines that attract other immune cells, like the killer CD8 T cells and natural killer cells, into the tumor.
So you're basically priming the battlefield.
Yeah.
Getting the initial troops in place.
Exactly. You prime the tumor microenvironment. Then, if you come in with something like a PD-1 inhibitor, it can release the brakes on those recruited T cells, letting them launch a full adaptive attack. The hope is this improves responses, especially in cancers that didn't respond to checkpoint inhibitors alone.
Fascinating. So what's the lead program in this area?
That would be KQB 168. It's their small molecule immunotherapy, currently in phase 1 trials for advanced solid tumors.
And how are they testing it?
Patients get the drug orally, either KQB 168 by itself or combined with Merck's Keytruda, the PD-1 inhibitor.
So testing that exact priming then unleashing strategy.
Correct. The goal is to see if KQB 168 can effectively prime the tumor, and then if adding Keytruda leads to better, more durable responses, particularly in patients who've already failed immunotherapy.
And is this immuno-oncology platform strictly for cancer?
Primarily, yes, but they have hinted at work in other areas, too. There's some preclinical data and patents suggesting they're exploring applications for their immune-modulating compounds in inflammatory and autoimmune diseases as well. So the platform might have broader potential.
Very interesting. Now, this innovative immuno-oncology platform must have attracted attention, too.
Oh, absolutely. Big attention. This led to more major partnerships. First up was Eli Lilly through their Loxo Oncology division back in July 2021.
What did that deal look like?
It was focused on the small molecule immuno-oncology platform broadly. Lilly paid $70 million upfront, with the potential for over $2 billion in future milestone payments. Lilly gets to pick promising candidates to develop.
And Kumquat's rights.
Kumquat kept commercial rights in Greater China, and importantly, an option to co-develop certain programs with Lilly in the U.S.
Why is a deal like that so important for a company like Kumquat at that stage?
Several reasons. First, it's non-dilutive funding cash infusion without selling more equity, which is crucial for an early-stage company. It lets them keep control and fund their other programs, like the KRAS work. Second, it's huge validation from a major player like Lilly.
Signals to the market that the science is solid.
Exactly. It builds credibility, makes it easier to attract talent, maybe even sets the stage for future deals, and it allows them to leverage Lilly's development expertise while staying relatively lean and focused themselves.
Okay, so that was Lilly in 2021, but there was another big one more recently.
Yes, just last year, April 2024, they signed an exclusive global deal with Takeda, this time specifically for KQB168, their lead IO drug.
Similar structure to the Lilly deal?
Broadly similar, yes. Takeda got worldwide rights to KQB168. Kumquat received significant near-term payments, potentially up to $130 million, plus over $1.2 billion in possible future milestones. Again, Kumquat kept certain participation options.
And Takeda's role.
Takeda funds and handles all the development and commercialization work beyond the ongoing Phase 1 trial, which Kumquat is leading. Kumquat gets tiered royalties on sales if it's successful.
So another shared risk, shared reward setup.
Exactly. It really highlights the perceived potential of this small molecule approach to complement existing immunotherapies like checkpoint inhibitors. Takeda clearly sees value there.
These partnerships seem incredibly strategic. They get funding, validation, expertise.
All while retaining space in their platforms and staying nimble. It allows them to advance multiple complex programs simultaneously without needing the massive infrastructure of a big pharma company themselves. It's a smart way to build value.
Okay, so wrapping things up, where does all this leave Kumquat Biosciences today? What's the outlook?
They're in a remarkably strong position, really. They've got clinical programs moving forward in both targeted therapy for KRAS and small molecule immuno-oncology. They've secured significant funding through these three major partnerships with Lilly, Takeda, and Bayer.
And they have that experienced leadership team with a proven track record.
Right. Liu and Ren know how to build companies, get deals done, navigate the path forward. The potential is huge. If KQB365 or especially KQB548 deliver positive clinical data against those tough KRAS mutants,
they could become a major force in oncology.
Absolutely. And if KQB168 proves that small molecules can effectively turn cold tumors hot and boost immunotherapy responses, that opens up a whole new treatment paradigm.
Of course, it's biotech. There are always hurdles.
Always. Drugging KRAS is still incredibly difficult. Competitors are right there with them. Mirati, Revolution Medicines, Astellas are all working on G12D inhibitors too. It's a competitive race.
And for the immuno-oncology side,
the challenges there are different, but just as real. You have to carefully balance activating the immune system without causing unacceptable side effects or toxicity. And then you have to clearly show that your approach adds real benefit compared to existing therapies. Plus, the regulatory landscape for combinations can be complex.
So significant challenges remain. But what makes Kumquat's strategy stand out?
I think it's their unique combination approach. They're not just a KRAS company, and they're not just an IO company. They're marrying deep expertise in covalent chemistry with innovative immune modulation, and they're doing it with the backing of strong partners.
Attacking cancer from two angles simultaneously.
Exactly. Using one platform to switch off the oncogene, the driver of the cancer, and the other platform to switch on the body's own anti-tumor immunity. The hope is that this holistic dual strategy could lead to much deeper, more durable responses than hitting just one pathway alone.
Which brings us back full circle to the name, the Kumquat. Small, resilient, packs that unexpected punch.
It really does remain a fitting metaphor, doesn't it? This company emerged relatively quietly, founded by these seasoned, brilliant scientists, and it's rapidly made a significant mark.
Their story is a great reminder, I think, that real innovation often comes from looking again at problems people thought were already solved, or maybe unsolvable, like an undruggable target, or maybe just a little citrus fruit with a surprising secret inside.
Well said.
Okay, that brings us to the end of this BioTale. Thank you so much for joining us on this exploration of Kumquat Biosciences. If you enjoyed it, please do subscribe, leave us a review. It really helps others find the show, and let us know what stories you want to hear next. You can find more info and resources on our website. Until next time, stay curious. And hey, maybe give a kumquat a try if you get the chance.